Welcome all to the today's class. Today we are going to discuss about urea cycle. After the urea cycle, we'll discuss the metabolism of lipids. So we begin with our code master MRCOG with concept oriented clarity. Now we are going to discuss the urea cycle. We know that the ammonia is getting formed in various processes, various enzymatic reactions, and this ammonia requires detoxification. And this happens in the cycle called as urea cycle. So this is the urea cycle where the ammonia will get detoxification process. Okay, so various things which are involved are the citrulline, you have arginocyanate, then you have arginine, and from this arginine you get urea, and finally the ornithine is formed. Okay, so you have ammonia that will get converted into carbamoyl phosphate, then citrulline is here, then you have arginocyanate. So we will see all these steps. So the first step is ammonia to carbamoyl phosphate. Then second step is ornithine to citrulline. Third one is citrulline to arginocyanate. Fourth one is arginocyanate to arginate. And from this arginine, via hydrolysis, you will get urea and ornithine will be formed. Okay, so let us see this urea cycle, also called as ornithine cycle or Krebs-Henschel cycle. What is the core concept? It is the detoxification of the ammonia which is required. Okay, so that leads to the formation of the urea in the process of detoxification of the ammonia. Now, what is the site? It happens in the liver. So remember, this again can be asked as a SBA. What is the site of urea cycle? It is the liver. Then in the cells, what is the site? Whether it is mitochondria, whether it is cytoplasm, where exactly? So it happens in the mitochondria as well as cytoplasm. Then it requires nitrogen, and there are two sources of the nitrogen. One is an ammonia, and second nitrogen comes from aspartate. Remember, the second nitrogen source is aspartate, and one is coming from the ammonia. So urea cycle is the only pathway for ammonia detoxification. There are two nitrogen atoms which are required, and they are coming from two different sources, one from ammonia and one from the aspartate. Okay, so what are the steps which are involved? This ammonia combines with carbon dioxide and forms the carbamoyl phosphate. That is the step number one which we have seen, and the enzyme which is responsible is the CPS1, that is carbamoyl phosphate synthase 1. It is present in the mitochondria, and this is the rate limiting step for the urea cycle. So ammonia combines with carbon dioxide, leads to the formation of carbamoyl phosphate. Enzyme is a CPS1 carbamoyl phosphate synthase, and it happens in the mitochondria, which is the rate limiting step. The next is a ornithine is getting converted into the citrulline. And here the enzyme which is responsible is a OTC, that is ornithine transcarboxy carbamylase. Okay, this is the enzyme which is responsible, and it also happens in the mitochondria. Here the citrulline exits mitochondria. Okay, so ornithine is getting converted into the citrulline. Citrulline is exiting from the mitochondria. The location is in the mitochondria itself. Okay, then you have citrulline plus aspartate will form the arginocyanate. Okay, but this takes place in the cytosol. So citrulline is getting exited from the mitochondria. The next process is getting formed into the cytosol. So citrulline plus aspartate will form arginocyanate, and the enzyme is responsible is arginocyanate synthase. Okay, so arginocyanate synthetase is the enzyme which is responsible. This uses ATP, and the AMP will be formed. Okay, now next fourth step is arginocyanate to the arginine plus fumarate. Okay, that is the step number 4. Arginocyanate is formed, and that will be converted into arginine plus fumarate under the action of the enzyme arginocyanate lyase. Okay, so this also happens in the cytosol, and this is linked with the TCA cycle. So this step is linked with the TCA cycle. Okay, and the final step is arginine will further form urea plus ornithine, and the enzyme responsible is arginase. This step also takes place in the cytosol, and ornithine is recycled. Okay, so the same thing we have seen. The first step is ammonia is combining with carbon dioxide, forming carbamoyl phosphate, and CPS1 enzyme is responsible. Then the formation of the citrulline is the second phase. Then here you get arginocyanate by combining with aspartate. The fumarate is getting converted. Then arginocyanate to arginine, and arginine to ornithine, and here urea is getting released. Okay, so these are the different steps, and these are the locations, that is either mitochondria or the cytosol. The first two steps will take place in the mitochondria, while the rest of the steps will take into the cytoplasm. So this is what is the urea cycle. So in the simple flow, you just remember ornithine, then the citrulline, then you have arginosuccinate, then you have arginine, and final you get a urea from the arginine. And here last enzyme is a arginase. Okay, so rate limiting step is the most important step. Here the enzyme responsible is a carbamoyl phosphate synthetase. CPS1 is the enzyme, and for this enzyme some activator is required, and the activator is NAG, that is N-acetyl glutamate is the activator for CPS1 synthesis. Okay, and overall this urea cycle happens in the liver. So please do not answer the kidney as a location. It takes place into the liver. Okay, so activator for CPS1 synthesis is NAG. That also you should remember. That is the essential activator for CPS1, that is N-acetyl glutamate. Okay, now there is a link with the TCA cycle. So this urea cycle, because it is linked with the TCA cycle, it is also called as urea bicycle. Okay, so urea cycle is linked with the TCA cycle, and hence it is called as urea bicycle. As like the reactions like fumarate to malate, malate to oxaloacetate, and oxaloacetate to aspartate is there. So there is a re-entry, it re-enters the urea cycle. So urea cycle plus TCA cycle will form the urea bicycle. Now let us see about the energy utilization and the energy production. So at the level of the CPS1, that is the rate limiting step, two ATPs are used. And at the level of the arginosuccinate synthase, again 2 ATPs are used, and the AMP is getting formed. So total you are getting 4 ATPs being utilized. Okay, so total 4 high energy phosphate bonds are utilized. Then you have some energy production also. At the level of the fumarate, you have NADH production. Yesterday we have discussed 1 NADH is equal to 2.5 ATPs. So gain is 2.5 and loss is 4 ATP. So net cost is around 1.5 ATP loss. Okay, that is the urea cycle energetics. Okay, now how this urea cycle is regulated? So it is regulated by the protein intake. If how is the dietary intake? And starvation. So that will lead to the increased certain enzymes. Then at the fine regulation is with the help of rate limiting enzyme, that is CPS1, which is activated by NAG, that is N-acetyl glutamate, and hormonal regulation is via arginine, that increases NAG synthesis. Okay. Now coming to the compartmentalization, we have discussed that this urea cycle takes place at two places. It is mitochondria as well as cytosol. If you see the mitochondria, that's first two enzymes, that is CPS1 and OTC, these two steps which are controlled by these enzymes, they happen in the mitochondria, while the rest of the enzymes are present in the cytoplasm. So after these CPS1 and OTC, rest of the cycle will take place in the cytoplasm. Okay, so remember first two steps are happening in the mitochondria, while rest steps are happening in the cytosol. Okay, cytoplasm. So there are various disorders of the urea cycle, and this is mainly because of the enzyme deficiency. So clinical presentation will be hyperammonemia, then encephalopathy, respiratory alkalosis, and patient may present with vomiting, lethargy, irritability, mental retardation, etc. So major enzyme deficiencies are CPS1 deficiency, second is OTC deficiency. Okay, so this OTC deficiency is quite common, and that is the only X-linked disorder leading to hyperammonemia. Okay, hyperammonemia is seen in OTC deficiency. Another is a CPS deficiency that will also have hyperammonemia. Then other deficiency is arginosuccinate synthase leading to citrullinemia. Then arginosuccinate lyase deficiency will lead to arginosuccinic aciduria, and arginase deficiency is mild in nature, and it will lead to increased levels of arginine. That is the final step where the arginine lyase will act on the arginine and the urea will be produced. So the arginine manganese deficiency will lead to increased levels of arginine. Okay, now let us see one by one. The most common is OTC deficiency. That is very common. It is X-linked disorder and it leads to the hyperammonemia, leading to increased ammonia levels and increased glutamate levels. Okay, so other is a ornithine transporter defect. That is a triple H syndrome. That is hyperornithinemia, hyperammonemia, and homocystinuria. Okay. So the management principle is that there should be low protein diet, adequate calories should be maintained, and arginine should be supplemented. Okay. So that is what is the management. Then let us see the summary table. So ammonia is finally getting converted into the urea. The site is the liver and the rate limiting enzyme is the CPS1. For this CPS1, you require activator, which is N-acetyl glutamate, that is NAG, nitrogen coming from two sources. One is from ammonia, second is from aspartate. There are four utilization of the 4 ATPs and net formation is a 2.5 ATP. Okay, because from the NADH, one NADH. Okay, disorder is a hyperammonemia and X-linked OTC deficiency is known. Okay, so last minute revision, CPS1 is the rate limiting, NAG activate cycle, two nitrogen sources, ammonia plus aspartate, 4 ATPs used, 4 OTC is X-linked and hyperammonemia is a key feature. That finishes the urea cycle, doctor. We have few questions. Dr. Sansi, can you please read the questions and try answering these questions on urea cycle? Yes, yes. Please go on.
Okay, sir. A 30-year-old man is being evaluated for metabolic disorder affecting ammonia detoxification. Which enzyme catalyzes the rate-limiting step of the urea cycle? Ornithine transcarbamylase, arginase, carbamoyl phosphate synthetase 1, arginosuccinate lyase, arginosuccinate synthetase. Sir, option C, CPS1.
Correct. CPS1 is the rate limiting enzyme. So CPS1 catalyzes the first step. It is the rate limiting and irreversible activated by N-acetyl glutamate NAG. So CPS1 is the key control point for the urea cycle. Next question, doctor.
Which of the following provides the second nitrogen atom in urea formation? Glutamate, ammonia, aspartate, alanine, carbamoyl phosphate. Option C, sir, aspartate.
Correct. This
Ammonia
also
and
we have discussed. Aspartate is the second source for the nitrogen. So urea contains two nitrogen atoms, one from ammonia and one from aspartate. Okay, we have seen two nitrogen atoms from two sources. So aspartate is the second nitrogen donor here. Next, doctor.
how many high energy phosphate bonds are consumed in 1 ton of urea cycle? 4 ATPs. Option C, sir.
Correct. So four bonds are utilized. Okay, it's been 2 ATP in CPS1 step and 1 ATP in cyclic AMP equivalent to 2 ATP. So total 4 high energy phosphate bonds are utilized. Next.
Which of the following correctly describes the location of urea cycle reaction? Entirely sir, option C. First two steps are in the mitochondria and rest all steps are in the cytoplasm.
Yes, because of the specific locations of the enzymes. Okay, so answer is C. First two steps are in mitochondria, rest is in the cytoplasm. Okay. So CPS1 and OTC are located into the mitochondria. Remaining enzymes are located in the cytoplasm. So two in mito, rest in cyto.
A newborn
Next.
presents with a newborn presents with lethargy, vomiting, and severe hyperammonemia. The disorder is inherited in an X-linked manner. Which enzyme is most likely deficient? Sir, option C, ornithine, OTC, ornithine transcarbamylase.
Yes, transcarbamylase. That is OTC enzyme is deficiency is more common. Okay. So ornithine transcarbamylase deficiency, most common urea cycle disorder. It is X-linked inheritance and causes severe ammonia. Okay. So from this again the same thing we are going to repeat. CPS1 is the rate limiting. Aspartate is the second nitrogen. 4 ATPs used. Second step, two steps mitochondria. Rest in cyto. OTC deficiency is X-linked. Okay. So that finishes the SBA questions on the urea cycle. Thank you.